Thanks for the opportunity to be here today. I wanted to thank Evan and Dana for two great talks. They definitely demonstrate what high-throughput, high-content imaging has come to be over the last 12 or 15 years. It's nice to see them again. We've all worked together at various companies. What I want to do today is talk about something a little bit different. And we're going to change gears here to talk about looking at cells in suspension. I'm with IntelliCyt, a company that has pioneered the use of suspension cells in screening. And what I'm going to talk about today now, as an overview here, I want to back up a little bit and tell you about the company, the origin of the company, and the technology, because it's not as well understood in this space as high-content imaging has come to be. Then we're going to talk a little bit about phenotypic screening in suspension and how enabling it is in some ways, how it compares to high-content imaging. And we'll go through a couple case studies. Some of our own application kits we've developed at IntelliCyt, as well as a few conclusions and some projections and some thoughts. All right, so IntelliCyt. Maybe I could just ask for a show of hands. How many people have heard of IntelliCyt? Oh, okay. Well, that's fantastic. It's a business that's actually 5 or 6 years old now. It's operated in a very efficient way, capital-efficient way, and it's developed a couple generations of instrumentation. Focused in the same area as everything you've heard about today, life science tools, focused primarily on bringing the power of a flow cytometry analysis to high-throughput drug screening. So we are using and leveraging a flow-like detection technology, but really focused on screening. The commercial status was as I mentioned, 5 years old. There's over 150 systems placed over 3 generations of instruments. The instruments have evolved in much the same way imagers have, from an automated microscope to these elegant, full-blown, high-content systems you can purchase today. We feel as though we've really accelerated a very similar process for suspension screening. Earlier this year, we raised a significant round of funding to scale up the company. And in doing so, we've grown quite a bit. Our unique position is bolstered a little bit by having intellectual property that's wrapped around a high-speed sample delivery to traditional flow. And we are the only company right now at this time that can deliver samples in small volumes at the rate that we do to use suspension cells and beads in a screening format. Very quickly, here's the evolution of the product. The IP is wrapped around the sampler. It was called the HyperCyt. That came out of the University of New Mexico. And the home base for the office is in Albuquerque. This can be bolted to a traditional flow cytometer, which means you can extend the capacity of the flow cytometers that are out there. There are many of these out there, and they're in use in a lot of core facilities. That evolved into the HTFC, high-throughput flow cytometry screening system, where a flow cytometer was integrated with the system and under control of a single piece of software. Quite a few of those out there as well. And then this year we released, with the help of some high-content imaging experts who've come over to the company, a benchtop, fully integrated screening tool that's set up to be automation-friendly. It can slide right into an automation pathway for cell-based assays, and it works with non-expert users. You don't have to be a flow cytometrist to start using this instrument within a day, and there aren't even any adjustments or knobs on the instrument itself, just a few things you have to do in the software. And this was introduced in January and formally released in June and is available now. As I mentioned, the home office is in Albuquerque. It's actually a great place to be for high-tech development with the Sandia Labs. Very good software development there. But what I wanted to point out is just this year, we've begun a San Francisco Bay Area expansion. So we are local now. I'm here in the Bay Area in business development. We have a marketing team here that's growing very quickly and local sales and application support. I don't think I have to go into these slides too deeply, but the meeting is focused on phenotypic screening. So I'm going to be showing you examples of the use of the technology in a phenotypic screening perspective. But the instrument does have a strong beachhead in antibody discovery. There's a lot of placements there for simply accelerating screening of hybridoma libraries, as well as target-based screens. Anything you can do in a cell that you can put in suspension. This slide, I think, since no one has showed it today, I'll go ahead and just mention there's been this resurgence of phenotypic screening because this is really the seminal paper by Dave Swinney, that they did a retrospective analysis of FDA-approved drugs between 1999 and 2008, and of the 259 that were approved, 75 were classified as first-in-class, meaning a novel drug, not a follower drug. And of these, a majority were discovered using phenotypic screening. So this is what sort of led the turn away from a target-based screening bias to a combination of the two. So we've heard a lot about this today. I've used all of these instruments over the years in a variety of jobs, but what tools do we have to do phenotypic screening now? And when you think about it, there's a lot of different techniques and a lot of technologies, but the three big categories are plate readers, microscopes, and flow cytometry. Plate readers are basically boiled down a population measurement into a single data point, and they can't gate out an irrelevant or individual cell, but they're fantastic for getting a lot of measurements done very quickly. Microscopes were sort of the opposite, and I think the advent of high-content imaging was that you could actually look at single cells, do the multi-parameter work, and reject artifacts within a population. And I think to date now, these instruments really do qualify as high-throughput screening tools. There are a couple limitations or overheads that come along with it. You're usually looking, you've seen lots of beautiful examples today, at pictures of 50 to 100 cells. And if you have to start taking more pictures per well, it starts taking a lot of time. But with the images comes a massive image analysis, data analysis, and image storage infrastructure that you have to buy into. No problem with that. I was involved in imaging for a dozen years. I think it's a fantastic tool, but this is just something that comes along with using this technique. Flow cytometry has great potential as a multi-parametric technique for cells in suspension, things that can be suspended, beads, the combinations of cells and beads, and it makes it quite versatile. It also can look at very large populations quickly, on the order of 10,000 cells in a second, which you can't do with imaging. So that's complementary to an imaging approach, provided you are not looking at a spatial information within a cell, but an intensity information or some characteristic of a surface expression or something like that. But obviously there's tens of thousands of flow cytometers out there being leveraged for fantastic cell-based assays. The problem has been speed and cost of the instruments and the ability to run through a compound library cost-effectively with the flow cytometry approach. One other advent of our platform is that you don't have to invest in the large image storage, image analysis, and data infrastructure component. And all the companies that offer that now do a great job with it, but it is just something you don't have to commit to. So this is the picture I'm going to show you because I will not have any other pretty pictures to show you the rest of the afternoon. It's my imaging envy photo. But this is a great photo, and I want to credit Joe Zock, who came from Salomics to IntelliCyt, with bringing this up, and I think it's a great way to take a look at something. We're talking about looking at a phenotype and an environment. And what you have here is a full ecosystem with a coral reef with adherent biology. You have suspension objects, all these tropical fish, and they all individually have independent phenotypes. And so if you think about how to screen, you're going into an environment like this, you need a model, you need a way to take a look at things in a way to think about looking at things that prefer to be in suspension in their natural habitat. Things that adherent should be looked at in their natural habitat. We have imagers, so we're focused on the suspension approach. Once you do that, there's a lot of power that you can leverage. You can look at subpopulations and multiple populations as they're dynamic and as they're doing their thing. You can look at a lot of things to find a rare event. It's harder with imaging. It's done with imaging. You can take a lot of images, but you can look at 10, 20, 30,000 cells in several seconds on a flow-based device, and suddenly there's quite a few of the rare events you're looking for. You can take a look at a whole population over time and look at how that population shifts and look at the dynamics of a population like that. So soak up the images because there aren't a lot of pretty pictures left. But now's a good time to talk a little bit about how the technology works. Essentially, I mentioned that you have the high-speed, low-volume sample delivery. This works by dropping a probe into the well that's under a constant suction. And when it drops in, it's pulling an aliquot out of the well of cells that are suspended by a shaking base for the plate. When it moves to the next well, it just goes up, over, and down. It draws an air gap. And as a result, each sample is separated by a small air gap as it goes through the tubing. This is really different than the way samples are delivered to a traditional flow cytometer, where you draw one sample in, it fills the whole line, you have a dead volume there, then you rinse and you follow with the next one. And even automating a flow cytometer is on the order of about a minute per sample. This approach, running in its fastest configuration, is 1 microliter per sample per second. It goes through into a very standard flow cytometry detection, 2-color laser with 6 measurements on the outbound, 4 fluorescent channels, and forward and side scatter. And then that gets analyzed and put into a software environment that very much emulates what you're used to seeing in high-content imaging. You have plate-based reading. You can essentially set up templates, set up an analysis, get a report, develop your IC50 curves, and so on. But the output is based on thousands of cells, single-cell data, and multi-parametric measurements. So, plate readers have obviously been validated for use in high-throughput screening and phenotypic screening. Microscopes have come along in the last 15 years, very well validated. And what IntelliCyt is here, I think, to prove and has done is really trying to bring flow detection technology on suspended objects into the high-throughput space. And just as a summary of the offering, it really is a complete solution. You know, the new instrument is designed to be a screening instrument. It comes with software that has multiple seats where you can look at your data off-site. It's essentially a database, but the data is so much easier to manage than images. It's not nearly as complex an installation. And then we're focused very heavily on developing our own kits, optimized for the platform, that are turnkey and amenable to screening to get phenotypic screens and other kinds of screens accomplished. This is just a very quick summary. I'll pull a few points here. So what does that mean really in speed? A minimal sample size is a microliter. It means we can read out of a 96-well plate in 3 minutes, 384-well plate in 12 minutes. You can do a larger sample. Say you're looking for a rare event. For each microliter, it's another second. It's a pretty linear increase in time. 10,000 cells second is typical if you've got the right density of cells in the well, which is about 1 million cells per mil. But in a 384-well plate, your sample size can be down to 15 microliters or so. So, not going to address this slide in detail, but these are words you see associated with all screening platforms. But I think this is one of the easiest ways to implement a high-content screening approach, provided your cells can be suspended. Now, it doesn't mean you have to be looking at blood cells or cells that are naturally in suspension to use the tool. You can resuspend adherent cells if it doesn't impact your biology. The other thing we've done in our own kits is really drive the cost of running these assays down to prices that are more consistent with high-throughput screening. And as a result, when we sell kits, we sell them in a 1, 5, 20, and 50 times 384-well plate kit. So that you can, and it's been automated, you can buy it with a robot. Take off my marketing hat a little bit now, but not entirely. I'm going to tell you first about our own internal kit development. Has been focused on a cell health and toxicity panel. So we're doing first toxicity, then there'll be cell function, but assays that are very important, as you saw in the first 2 presentations, to general phenotypic approaches. Okay, so both Dana and Evan talked about toxicity and measurements in phenotypic screening. The important thing is our internal group has taken these relatively well-known assays, optimized them for the platform, made them addition only or no wash, mix and read, whatever terminology you want to use. You're essentially just adding the probes to the wells some time period before reading. The cells are in suspension. Washing can be a little trickier with a suspended cell line, right? So these assays are developed to avoid having to do that. We've also collaborated with Lightron Corporation that's developed a certified and accepted genotoxicity test as part of the toxicity screening you would do with compounds. I'll tell you a little bit about that. That's a really interesting test. It deserves more time, but if you're interested, we can certainly get more information. We've even begun to experiment with multiplexing cell lines on the instrument. So if you have a cell line expressing a species-specific antigen and you want to test it for species cross-reactivity, you could put mouse, rat, monkey, human, 4 cell lines, multiplex them with colors, and run them all in the same well. So there are applications for that, which I won't talk about today, but everything about this platform is to get more data more quickly, in a multi-parametric way, cost-effectively. And then I'll finish up talking about a phenotypic screen run by one of our customers for acute myeloid leukemia. And I think a very elegant way that he evolved the screen to run on this platform, because in this case, this was the right platform for the screen. So let's start with cell cycle. We talked a lot about cell cycle today. I don't need to go into it. On a flow cytometer, the sort of gold standard dye has been propydium iodide. But when you think about screening, it's not the best probe. It requires you fix the cells. It requires they be permeabilized and that you treat with RNase, because it also stains RNA. What you're looking for is a doubling in the total DNA content, so you don't want the RNA contamination. So that's supposed to say washes. All right, so this signal also bleeds into some of the other output channels. Why do we care about that? Because we want to multiplex cell cycle with other channels. You might have a GFP in there doing something else, or you want to use your other color channels. So we went looking for and found a probe that's very specific to DNA. You can stain live cells. You remove the requirement for fixation and permeabilization, and you don't need the RNase. Okay, so the dye goes right in. Pretty selectively binds the DNA here. No contamination from RNA staining. You don't have to work with a fixed cell. So you can be doing this on live cells. Second kit that's out and validated, these were all just released in June, is a 4-parameter apoptosis kit. So we're able to multiplex here with a cocktail of dyes that mark caspase activation, mitochondrial dysfunction, phosphatidylserine translocation, and ultimately cell death. This gives you a fingerprint or a phenotype of the apoptotic induction as a result of compound treatment. What's important about this is that you don't end up missing an important step if you do the assay at the wrong time. You get a more complex fingerprint, which can be useful. Obviously, it applies across the entire spectrum of drug discovery, toxicology. It's a very important bread and butter assay in the industry. So what we've done here, again, it's a no-wash assay. It's a cocktail you add to the cells 1 hour before you read the plate. Just add it and read. You get 4 simultaneous readouts. You haven't missed an important event, and the protocol is quite simple. For genotoxicity, there's the gold standard, and it's a very labor-intensive assay, has always been microscopy. And when you're looking at the genotoxic potential of a compound, and that is to damage the DNA, which creates potential, obviously, mutations and carcinogenicity, you're looking for these things, these little satellite pieces of DNA that are called micronuclei that come off of a cell. It's complicated because there is also blebbing, there's other things there. In my time at Molecular Devices, we spent a year developing an algorithm to accurately measure micronuclei and ignore other things that you see in imaging. Originally, this assessment was done manually on microscopes, people looking through thousands of cells. It's a rare event, so it can be 1 in 100, 1 in 1,000 cells, depending on the potency of the compound. But it's absolutely something that needs to be ruled out in the drug discovery process. Automated imaging did a fantastic job of bringing a potentially biased but also labor-intensive method down from days or weeks to 16 to 18 hours to run a single 384-well plate. And the reason it takes that long, the imagers are very fast, but you have to take a lot of images to get enough images where you see the rare event. Once you start looking at 10,000, 20, 30,000 cells per well in a flow-based technology, you can bring that down to about 30 minutes to read a 384-well plate. So now you can talk about doing this micronucleus induction assay on a lot more compounds cost-effectively, possibly even earlier in the process. which could be very valuable. You don't necessarily want to lose these compounds. You may want to modify them and make them non-genotoxic. So this assay was developed originally by Litron. It's been accepted as one of the cell-based assays for genotoxicity formally. And I want to tell you just in one slide kind of how it works. If you want to learn more about it, please contact us. It's a two-dye assay. So if you have cells going through this kind of treatment, you end up with necrotic cells, apoptotic cells, and we use one dye to identify that. So the red dye is going to identify dead and dying cells and necrotic cells and the small blobs of DNA that result from that could be mistaken for micronuclei. And then the green dye comes in specifically in healthy cells and it's going to label the DNA in the nucleus and the micronuclei. And then the adherent cell line you're doing this with is lysed, and everything comes into suspension and is run through the instrument, and you're actually counting all of the objects, not cells in this case. You get a quantification of the micronuclei that are green and the nuclei that are green, and you can normalize micronuclei per cell number. It's a different approach to the assay, but it's very robust and uses much larger populations and much, much faster. Quick table to compare the two, and this is an application where I think both technologies work. They work well. If it was my lab, I would probably do a lot of compounds on a flow-based platform. You can get through the plate in, say, 30 minutes. Look at a lot more events. You're going to generate a lot less data. We're talking about megabytes here, 70 megabytes compared to gigabytes of images, and then take a subset of those compounds, reconfirm them with an imaging approach. But this is a way to think about doing your genotoxicity cost-effectively, more quickly, on a lot more samples. All right. So now let's talk a little bit about a screen done outside of our hands, work done outside of our hands. This was work that was presented at a user event, and David is interested in acute myeloid leukemia. As a disease, it's problematic. You have a mutation that presents the normal development of blood cells, and immature cells are accumulating in the bone marrow and not maturing, not exiting. And the 5-year survival rate of this leukemia is about 25%. The therapeutic strategy they wanted to employ is just kick those cells into the mature state, get them going, right? Get rid of that phenotype. So we're looking to change the phenotype of the cells. The problem with the assay is the model systems are not good for screening. There isn't really a good screening tool to do this. There's some elegant ways to see the changes in the cells, but none of them were practical for screening. So he evaluated a few approaches and came up with, the key idea was to put a promoter, to create a cell line, a murine cell line with a GFP expression under the control of a promoter that only turns on when the cells mature. So the cells turn green when they mature. Now you've got a very easy way to see mature cells, right? So they took that and they decided now to develop a way to screen that cell line. So what's the best way to do this in high throughput? Some of the traditional methods, light microscopy, is very clear. Fluorescence microscopy with a functional assay, you can stain, you can see the mature cells are quite phenotypically different. Flow cytometry. You can stain surface markers and you can see big shift in the change as they mature. No problem. But how do you do this in high throughput, cost-effective way? With this marker, if you look at a flow cytometry plot, here's the immature cells, mature cells. As they mature, you drive the population into the green expression. The screen was run with 384-well format. These are suspension cells. Remember, they're non-adherent. They're added to each well, compounds in each well, 4-day incubation, and then the evaluation as to whether or not we have a shift in the population to the GFP expressors. So they tried imaging. They had a little bit of trouble with suspension cells and imaging, which is typically the case, right? You can get okay images. They had edge effect problems. Cells were migrating towards the edge of the plate. Cells pile up on top of each other. You could still do an assessment here, but it was determined that it's not, with the cells not being evenly dispersed and overlapping each other, it wasn't ideal. And also another probe would have to be added to assess cell viability. So this is what they decided to do. They take the cells, they've got the progenitor cells, They treat them with compounds. They look for green expressors on the high throughput flow. This was done prior to the release of the IQ, so it was done on the predecessor instrument, the HTFC. The nice thing about forward and side scatter and flow is you can actually use those channels to distinguish cell viability without adding another probe. So now the first step in the analysis strategy is to gate only on the live cells, then gate on the green cells. It's that simple.